Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntazir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic. So during this topic, we will be talking about X-ray crystallography. That is a technique for understanding the molecular and physical structure of a crystal. That crystal could be of any other molecule, such as a chemical compound, any organic chemical compound, including macromolecules of all the life present on Earth planet. For example, proteins, nucleic acids, or other organic compounds. With the help of this technique, we can identify the structure of those unknown proteins or nucleic acids. For example, the DNA, the deoxyribonucleotides. That DNA structure is also derived by using X-ray crystallography. At the very early point, that gives us an idea about how exactly whether the DNA is spherical or like a spiral. In this way helps us to find out the structure of DNA. Notable point that the overall X-ray crystallographic process is carried out for unknown proteins. In this lecture, we will see what is the basic concept of X-ray crystallography and what is X-ray crystallography. It is performed only to crystals because in this case only X-rays are used instead of any other light, because we can't use normal radio wavelength light, the visible light. That's why we use X-rays only, because we are dealing with the molecular structure here, the structures in the very atomic level that cannot be determined at all with the visible light. Therefore, we need to use X-rays. Next important question here will be why we use crystals for crystallographic process. In any molecule, if we want to see, we need to put it into the crystal format. Crystal format is generally obtained by cooling and condensing the compound solution for a normal compound that we are trying to find the structure of. Now, why we consider the crystals? Normally, all those molecules such as organic compounds, all those macromolecules, whatever we are talking about, those molecules are not stagnant. They are present in different orientation, which means they are moving, they are rotating, the bonds are changing with the time, and all those things are modified from time to time, because that is for the living organism, that is true. They are changing over the time, but it is very difficult for us to identify a molecular structure if all those bonds are changing, rotating and modifying. So the only way and easy way to make the structure is to make that whole molecule freeze, fixed in a point that we can achieve a crystal of the molecule. Once we make the crystal, we can be sure that molecules that we made the crystal must be fixed, permanent and stagnant. Now once that molecule is stagnant, they have different type of atoms linked with that in different regions, and that will be fixed throughout the experiment. From the beginning to the end of the experiment, we use the same region of that same atoms that are present in the same region of that crystal. So it is very important, very easy for us to identify that target molecular structure. But for those types of molecules which we cannot convert into crystals, we cannot use them into X-ray crystallography. Now let's start with crystal structure. Crystal means we are discussing about 3D structure. Suppose it is a protein molecule that we are going to discuss in the three-dimensional space. There is a protein structure in it. Now we see the main and basic idea of X-ray crystallography. That is, we put X-rays to interact with this crystal. Whenever we apply X-rays to this crystal will diffract the part of this X-ray. Diffraction means very similar to the reflection, during which it simply changes the direction of angle. When X-ray hit to this crystal, then it is going towards the crystal interacting with the crystal, and then it is diffracted in a specific angle. Okay, it depends upon the type of probe molecule. When we are analyzing the diffraction pattern, it varies, and this diffraction pattern is unique for different types of molecules. If the molecule is more complex, then there will be more different varieties of diffraction that we will learn later. And if it is a simpler molecule with very less regions, that are most regions of the crystal, if it is blank, then it will give us less diffraction. That is the complete idea, from which we will achieve informations from the diffraction, and that informations will be helped us to know about that molecule as the structure of that molecule. If we are identifying the structure of a protein, we have to make the crystal of that protein. Suppose this is a protein crystal, not a real geometrical shape, but only a rough part of protein crystal. For it, very common idea is we are bringing the x-rays for applying to the crystal. Then these x-rays are diffracting. We also have a detector and x-ray film at the end on which we can see the spots after the diffracted ray. So first diffraction visible here, next here, next here, and next here. So there are different diffractions. When we see this x-ray film, we will see the different diffraction pattern. Whenever these x-rays are hitting to that film, we will have different spots there, here and there, that are generated in that film by the diffraction of x-rays. That plate is very important to search for, to find out, to find about exactly what kind of protein structure that we are considering for, because a protein is very complex molecule, macromolecule. It is made up of so many groups, so many subunits, and many things are attached with each other in it. When we consider this three-dimensional shape, the idea is X-ray will come and will be diffracted. That means there should be a plane for the diffraction. If we study about diffraction in overall process, it means it is very similar to reflection. Suppose there is a ray, light or wavelength comes in. It hits this specific plane and reflected away. That's the judgment notion. So the diffraction is due to different planes in the crystal. Okay? So what we do is we take that crystal and we split it down into smaller unit fragments. Students, keep your attention to this important topic. That what am I saying? Suppose we take this crystal, the whole protein, and break them down in smaller fragments that are imaginary fragments. We are not actually breaking the crystal here physically, but I say we are only breaking the crystal in imaginary fragments, imaginary units, or in imaginary parts of crystal for understanding you. Now we take each of those imaginary unit of the crystal. Now we will not take the whole crystal, but only a smaller part, a smaller fragment. And if we zoom in here, one of such smaller fragments gives us structure like this. This is a zoomed in portion of a smaller unit fragment, and this unit fragment has some portions of proteins. So these three red colored regions are the portions of proteins, fractions of proteins present in the part of this small zoomed fragment, right? So now what we do is, the answer is that we are trying to find out different planes in that unit cell. Because if we take it as a cube, as it has a cubic structure, there will be three possible things: H, L and K. Height, length and K. So these three things you can easily put it there. Like height will be here, length here, and K will be in that region, or whatever, you can take it as your own choice, as there are these three things that you will be requiring to find out the actual structure of this cube, geometrical shape, explanation or idea about that cube. For this purpose, we will try to find out different planes of each of the single unit. For example, we are trying to find out a plane that is present in between this region. It will be a kind of hard square shape. It is for you to understand about this plane because it is not a 3D board but 2D. I mean the picture is a 2D region. I cannot draw a 3D structure in this board. Now we try to imagine this is a small cube, and if we are rotating the cube in a way so that we find a specific plane. In this case we are dissecting this H side to get a plane. Okay, so we are placing it in a way so that we get a plane of diffraction. Then X-rays come and hitting to that plane, and then these are diffracted. Suppose this is the plane that we are talking about. This is the X-ray film and this is the X-ray. We have a diffraction here. For example, now the diffraction is calculated from the distance of the center of axis. Center means the straight. If there is no diffraction and UV passes by wherever it gets hit to the X-ray film, that is the center, and all the distance is calculated based on the distance from the center. So here the first diffraction is at this point. Let's say this is the first diffraction from the plane number 1. Actually they have different number of planes based on how we dissect the diffraction lengths. So in this case the plane number 1 will be 200. From this plane number 200 we get the diffraction at this specific point only. Now we do the same thing. We change the plane number 2 to 0 in its plane. We have a diffraction. For example, it is a second point of diffraction. So by this fashion, what we do? We take different diffraction planes here and we trying to find out the diffraction pattern. Not only the diffraction pattern, but we also need to think of two things here. So when we are analyzing the diffraction pattern, the first thing is the angle of diffraction and the second thing is the intensity of spot. Why? Because the intensity of the spot that is going to form in that X-ray film is directly proportional to the electrons present in that plane. Now let's assume the diffraction pattern is very bold here at this specific point. That means the diffraction formed in this region, the plane with which the diffraction formed in this region contains a very high density of electrons. The very high density of electrons means there is very high chance of finding of the content or the protein molecule for which we are considering. So higher density of electrons means if we go back there and see that plane, there must be most of the protein components present in that plane. Whereas if this intensity is very faint, very low, it means there is low electron density present. Low electron density means there is a less chance of finding the fragment of a protein in that plane. Now if you analyze this cube, that is a unit cube, if diffraction took place here from this specific plane, here an important question comes in our mind that what we will find. Huge amount of protein is present, so we will derive higher intensity of the spot. So higher intensity will help us to identify, yes, there are high amount of protein or protein content present or maximum amount of electron density present in this region. Now if there is a diffraction from this specific plane, from this specific part, and if I put a plane like this and from this specific plane that gives us a very faint spot and tell us that there is a less chance of finding a protein component there. So now try to calculate the complete thing and going backwards. We know that the amount of intensity corresponds with the amount of electron density. With the help of many experiments, scientists have admitted this idea that if we have this amount of electron density, it corresponds to the 10,000 electrons. If we have this amount of electron density corresponds with the 6,000 electrons. We know about the relation of number of electrons associated with the intensity level that we find in the X-ray film, as we know that we are dealing with the structure of unknown protein. We simply derived that spot based on the density and the angle of diffraction. We have not talked about that yet, but based on that intensity we can identify where and which part of the unit cell contain more electron density area, which part contain less electron density area. And this angle of diffraction is also very important because this angle of diffraction is different for different planes. As we are changing the place, memorize, we are not changing the association of the protein molecule because this protein is fixed, it cannot rotate. Those atoms present in crystalline form. So as we are changing and altering this plane, we are getting different diffraction angles. So diffraction angle is also telling us about how exactly we are placing the plane. So this diffraction angle will tell us about which area of the region we get the spot for. So it will be important for the computer to find out which spot represents to the which area of unit cell, and intensity will be helpful to find out the amount of electron density corresponding with the area of the unit cell. As we know, both these things together for one small unit cell, and all of the data is feed into the computers. Now go back as we divide the actual crystal. Let us say 3 different small units. We get the data about each unit for all these 300 units, and then we compile the whole data for 300 units, which will give us an idea about the whole three-dimensional cube and the different density of the electron in that cube. So as far as we go with this protein, we will find high electron density present throughout this type of area, which is going to help us identify and understand where exactly the protein molecules and different atoms are present. But it is not end, because it is going to give us an idea about where exactly the components are present. It is not going to give us the exact structure of protein. So how could we get the exact structure? Now we correlate the data of the electron density along with appropriate structure of the protein. As a protein is consisted of its secondary structure, that are alpha helix or beta sheet. There could be better turn and loop. So we know again with the help of series of experiments, we know that which kind of electron density map corresponding with the alpha helix and which kind of electron density map is corresponding with the beta sheet. So with the help of this correspondence an idea, we can analyze that what kind of and where exactly the alpha helix is present in this cube, where exactly the beta loop is present in this loop. That is going to give us an overall idea, an overall view about the protein structure. Now that is also not the end. That is again a crude extract an idea. Now we take advantage of the other type of computerized biotechnology tools, and like the bioinformatic tools, we take the protein data bank. We take the same type of protein structure. We compare this structure with each other together to find out exactly where there is a difference, a change, that we accomplish the gap from the idea and the informations from the database. But that is not so, not also the end, because it is a structural part. We also try to perform several experiments to find out the function of unknown protein. Once we know the function, we can correlate the function with the structure to get exactly what kind of structure the protein should have to obtain that kind of a function. Because if we try to know the functions, we can go back to get a structure. If we get the structure of a protein, which will help us to determine the functions of this specific protein, because there is a correlation between the configuration and function of the protein. So that in a sense we have known that how correctly we derived out the structure of the protein or any other molecule with the help of X-ray crystallography, that is based on the X-ray diffraction pattern. And we think there are two implements that I have not talked about till now, which are very important. First is Bragg's law and another one is a Fourier analysis of Fourier transformation. Both these cases are mathematical annotation, which I am not going to replenish and puzzle you. But the idea is whenever there is a diffraction going on from the nearby range of the electrons, so nearby range of the atom of the molecule, that we are looking at diffraction, the wave of the diffraction could be in two different ways. Now it could be in the same phase. Let's say it is coming from two consecutive area of the atom of the molecule. We have both of them in a phase. So if both of them are in phase, the total idea is constructing. So it is called a constructive interference that is going to enlarge this whole wavelength, and it is going to give us a brighter spot here, a brighter light here. Now if it is coming from two different zones, two different regions, and if those light or rays are not in phase but they are out of phase like this, they strike out each other, cancel each other, we call it as a destructive interference, as it gives us a faint band. It is also very important whether what kind of nearby electrons that this light is or this X-ray be sprinkling, affecting and scattering. Due to this sprinkling and scattering, what kind of band we're inspecting, but based on this band intensity and angle, we finally obtain the whole confirmation, whole design by extrapolating the data and also tell about the data with the help of experimental decision. Okay, so it was our topic today, X-ray crystallography. I hope you are truly understanding the overall point of view about X-ray crystallography. So students, it is my second video that has questioned me by my one student. How you found it? Please tell me in comment section. If you want to ask about any other topic about science subjects, then you can.